Good afternoon. It's a pleasure to present to you today some work we've published on tumor-like behaviors of smooth muscle cells and atherosclerosis, as well as some unpublished emerging work also. My objectives today are to briefly review the genetic contributions to coronary heart disease, and then to present some aspects of tumor-like features of smooth muscles and atherosclerosis, and in the context then explore novel therapeutic concepts and opportunities. I bring this talk in the context of the following perspectives. Firstly, that residual cardiovascular risk on Ldlr therapies remains the major cause of death and disability, so this is an unmet clinical need and therapeutic opportunity. Human genetic studies have revealed hundreds of loci for atherosclerotic cardiovascular disease, and therefore human genetics can drive our mechanism-based predictions on therapeutics. Third, most genes that cause coronary heart disease at these loci have been yet undetermined, and the gene has not been identified, and the function and the mechanism is unknown. And last, from enrichment analysis, we know that genes at these loci are markedly enriched in their expression within stromal cells like smooth muscle cells in the vessel wall and other vessel wall cells also, but pointing us back to the importance of the vessel wall and smooth cell biology in human cardiovascular disease. In that context, human genetics has revealed over the last 20 years over 300 loci for coronary artery disease and myocardial infarction at genome-wide significance. Most of these do not relate to traditional known risk factors pointing to novel vascular mechanisms. Most of these have unresolved mechanisms, functions, and an understanding of the cell or tissue involved. However, we can glean knowledge from some of the genes that are enriched at these loci. Many loci contain genes that are enriched and expressed in vascular smooth muscle cells and matrix, like ADAMTS7, which I'll talk about in a moment or two, but others also. In addition, there are genes that are enriched within endothelium at these loci for inflammatory pathways. And in general, we're able to understand more about pathways from the genes that are enriched at these loci, but it doesn't create causation or knowledge of mechanism. We are also now knowledgeable of an unexpected role for somatic mutations, which has arisen from human genetics over the last decade, in particular clonal hematopoiesis, with bone marrow-derived cells being recruited to the blood vessel wall and driving abnormal inflammatory activity in the blood vessel wall. Now, in 2011, we published our first US study, which revealed a couple of loci for coronary artery disease and myocardial infarction. I highlight here ADAMTS7, which was a locus for coronary atherosclerosis and for myocardial infarction. This has been highly replicated locus through the Cardiogram plus C4D consortium and others over the last 15 years. And we've done a significant amount of additional work on the biology behind this. Still remains a bit of an enigma, but I'll share a little bit of that with you. We know that ADAMTS7 is highly expressed in smooth muscle cells and in endothelial cells. And we also know that in vascular injury models, in carotid or femoral artery injury models, that a knockout in a mouse model of this gene prevents the response to vascular injury and the stenosis or restenosis that we see in that setting. In addition, shown here in the ApoE atherogenic mouse model, knockout of ADAMTS7 blunts the development of atherosclerosis both in female and male mice, both in the aortic on face and on the roots. And this is true also in the Ldlr model. So in that context, it suggests that ADAMTS7 is at a locus for coronary artery disease, and that knockout or loss of function of ADAMTS7 in mouse models of atherosclerosis is protective. However, it remains unclear from human genetics because of complex pseudogene genomics at this locus whether loss of function or knockout of this gene in a human is protective for coronary atherosclerosis, and this is limited therapeutic development. Again, speaking to the importance of human genetics to resolve causality and directionality in therapeutic development. Now, in this context, about 8 years ago, Japan joined my lab and was really anxious to use mouse models with very advanced lineage tracing and single cell technologies at that time to profile the changes that occurred in smooth muscle cells and smooth muscle-derived cells in atherosclerosis. In other words, what types of cells were coming from smooth muscle cells and where, and how do they transdifferentiate into other cell types, and could you manipulate these cells and trace them using single cell technologies and lineage tracing? In a tour de force that Huize published in 2020, he used single cell technologies and lineage tracing to show these remarkable diversity of cells that are generated from smooth muscle-derived cell. And we can see here a population of cells called SMCs, which are not the same in transcriptome profile, single cell profile as smooth muscle cells or other cell types. And these SMCs are, however, lineage traced to be green or ZS green, smooth muscle-derived cells, as we can see here. When you compare the transcriptomes, they have downregulation of smooth muscle cell gene markers, contractile markers, but upregulation of some inflammatory cascades, matrix components, but importantly, several genes involved in monocyte, endothelial, and stem cell biology. So we call them SMCs at that time. In summary of that work, we found that SMCs were derived from smooth muscle cells through transdifferentiation and could go on to form more fibroblast-type cells, lipid-laden foam cell-like cells, or could indeed be pushed backwards to our smooth muscle cell-like phenotype. So they were fairly plastic. At the same time, we found that retinoic acid signaling regulated this pathway, and blocking retinoic acid would modulate this and reduce the amount of inflammatory cells and cells derived from smooth muscle cells in the lesion. Tom Gutermus's lab, Gary Owen's lab, at around the same time had really revealed the importance of KL4 and OCT4 for Gary Owens' group to regulate these intermediate cells, and for TCF21 to regulate the formation of these intermediate smooth cell-derived cells and fibroblast-type cells. Huize took this further to ask questions about more unknowns at this point. What were the mechanisms? What were the features? What was the behavior of these cells, these intermediate cells? Again, using mouse models, he focused on certain areas that he had discovered in his first set of experiments. He found that smooth cells have a significant amount of DNA damage in his preliminary data, and from published work we knew that they also exhibited clonality in both human and mouse atherosclerosis, and this was known for over 50 years, in fact, but had been largely lost in the literature or ignored in the literature over that time. It's also been true that the question of whether atherosclerosis is a tumor-like disease has been raised, particularly by Jeff Ginsburg, over 20 years ago, but really no systematic studies were performed, particularly in the realm of smooth muscle tumor-like behaviors. So Huize took this on. The first thing that he did was he demonstrated that smooth muscle cells marked in ZS green from lineage tracing here early in Western diet feeding Ldlr deficient mice have evidence of oxidized DNA damage before the formation of lesions in the intima. And that over time you see early oxidized DNA damage followed by the formation of the intimal lesion, which is smooth muscle cell rich in the early instances and then becomes more complex. Looking at mouse material from a single cell perspective and using computational approaches like copycat, he was able to infer the copy number variations, DNA copy number variations, gain of copy or loss of copy across the genome, and compare to a reference of healthy smooth muscle cells in diseased blood vessel wall, the intermediate stem cells, and other smooth cell-derived cells. And you can see that there's an increasing in red and blue throughout this process, which suggests an increase of gain of copy in red, loss of copy in blue, across multiple areas of the genome in mouse atherosclerosis over time. This was not unique to mouse, and you can see profound evidence for gain of copy CNVs and loss of copy CNVs in smooth muscle cells from atherosclerotic lesions in humans compared to controls. This is one of 12 lesions we examined this way with more than one technology for predicting copy number variations. Interestingly, the gain of copy number variations typically occurred in areas which we could find both GWAS coronary artery disease loci genes and cancer cell genes, and I'll come back to that a little bit later. In addition, when you isolated these cells using ZS green and some of the markers we had described for SEMS, and then cultured them versus smooth muscle cell-derived cells that were not stem cells. And the smooth muscle-derived cells had more evidence of proliferation at early passages and remarkably had sustained proliferation over 50 passages shown here, which was not true for smooth muscle cells without these features, which died off around passage 10 or 12. In fact, these cells continue to grow to hundreds of passages in our hands. In addition, these cells also in culture demonstrated a resistance to senescence in the smooth muscle-derived cells, even out to passage 50, compared to normal smooth muscle contractile type cells that were derived from the blood vessel wall, as marked by beta-gal staining here. And this was associated with loss of P21 and P16, which may be responsible for the protection from senescence. And in lesions, we found that compared to young, the media of aging mice developed senescence, as shown here. And in atherosclerosis on Western diet, also the media developed senescence, but there is a loss of senescence in the smooth muscle-rich intimal lesion here, suggesting that the smooth muscle cells in the intima of the lesion, in fact, smooth muscle-derived cells lose senescence capacity over time. Huize went on to show using software for cancer pathways, focusing specifically on cancer activation pathways, that multiple cancer activation pathways, except for TGF-beta, in fact, were turned on in all of the smooth muscle-derived cell types compared to smooth muscle contractile types. TGF-beta is involved in generating contractile smooth muscle cells as well as response to injury, so not surprising. So multiple pathways were activated using single-cell data analysis, and then we confirmed these using phosphoantibodies for the phosphorylation state of markers of each of these pathways, and that's shown in the paper. So in summary, Huize had shown at this point that smooth muscle-derived cells in atherosclerosis have evidence for extensive DNA damage and genomic instability, and I showed you one example, but we showed that in a number of different ways. They have sustained proliferation and resistance to senescence, and not shown here, they also form colonies and invasive capabilities, which normal smooth muscle cells in cultures do not, and they had cancer stem features, which normal smooth cells do not. And there was evidence for activation of multiple cancer-associated pathways. But is this important, or is it just an epiphenomena that this happens with the disease and as a result of the disease rather than leading to the disease or contributing to the disease? So Huize developed a couple of experiments to assess this question. The first was there was a mutation that's common in tumors, a KRAS G12D mutation, and KRAS was one of the genes that's highly expressed in smooth muscle cells and in endothelial cells. It was found to have gain of copy number variations in our copy number variation analysis. It was found to be in a pathway activated in our pathway analysis. And it is found to be overexpressed in lesions in atherosclerotic plaques. So Huize generated a mouse that had KRAS G12 specifically overexpressed in smooth muscle cells and then crossed the Ldlr receptor and then put on Western diet. And as you can see on the bottom two panels here compared to the top controls with the mutation, there's a marked acceleration in early development of oxidized DNA damage and in early lesion intimal development over time compared to the controls. And this is shown pictorially on the left and quantitatively on the right with oxidized DNA damage, evidence for intimal smooth muscle-derived cells, and then lesion area and lesion size. And when we stage these lesions for complexity and maturity, they're markedly more mature, markedly advanced with the KRAS mutation. In contrast, we were also able to use therapeutics that are available in clinic for cancer to manipulate DNA damage repair pathways to block the actual development of atherosclerosis, block the migration of the smooth muscle cells, block the formation of the lesions and oxidized DNA damage, providing proof of concept that smooth muscle cell mutations in atherosclerosis accelerate disease, and blocking the behavior of these cells can limit disease. And this introduces the concept of atherooncology, where smooth muscle cells with DNA damage and instability can lead to progressive and accelerated disease, and manipulating that may become a possibility for therapeutic targeting. And this adds to our information that clonal metapoiesis in cells outside the blood vessel wall from the bone marrow and the blood that come into the blood vessel wall with somatic mutations also can accelerate disease. This is particularly true in an aging population. And this shifts our thinking towards thinking of arterial wall targeted therapies, regardless of LDL cholesterol level, that could suppress general activation of tumorigenic pathways in smooth muscle cells and perhaps in myeloid and other cells. And again, thinking about precision medicine targeting patient-specific genetic characteristics. What kind of mutations are occurring in the lesions of this particular patient? Which therapy could be chosen to target those mutations in that behavior? And this is particularly an ongoing discussion in clonal hematopoiesis, for example, but could be true in other cells in the blood vessel wall, as suggested by our data. I'm going to switch in the last few minutes to a little story that we're developing around human-based discovery of genes related to smooth muscle cell behavior and atherosclerosis. And this is a collaboration across multiple labs and our team of Enrique, Sharif, and Thomas who've been involved in this work. So the Pakistani genomic resources is very large, now over 250,000 participants recruited in Pakistan to studies of complex disease, particularly cardiometabolic diseases led by Dinesh Salehin. And the reasons of huge, powerful resources that about 40% of first cousin marriages in Pakistan are first cousin marriages. And this leads to a marked enrichment on homozygous knockouts and heterozygous knockouts at the population level because of recessive inbreeding. And this allows us to study loss of function mutations at much higher scale than elsewhere in the world because of this enrichment. Using this resource, I'll describe the process of some discovery that we've done. But first, we took all of the genes that were at GWAS loci in a recent million veteran product GWAS study, and we filtered these by narrowing the focus to 1 megabase at each locus. We filtered them further by getting rid of all known lipoprotein GWAS genes because we're not interested in those at this point. It was the vessel wall we were interested in. And then we went on to actually filter these further by enriching for expression in smooth muscle cells using bulk RNA sequencing data of atherosclerotic lesions, GTEx and the like, our own single cell profiling of human and mouse atherosclerotic lesions for differentiation highly expressed genes in smooth muscle cells in atherosclerotic lesions, and also using EQTL data for smooth muscle cells in culture. And this allowed us to narrow the focus down to about 250 genes. At that point, we performed an experiment getting back to the Pakistani genomic resource where we asked the question, at these loci that contain these 255 genes, was there any of those 255 genes that had a strong signal at whole exome sequencing, whole genome sequencing for coronary artery disease? So we put together a resource of over 100,000 cases, about half of them the controls for coronary artery disease, myocardial infarction with whole exome, whole genome sequencing data across the resources described here. And we discovered which genes were top of that list from the 255, suggesting that that gene at that locus was probably the causal one, because when you have a loss of function associated one gene at a locus, it's most likely the causal one for the GWAS signal. And here's the top 10 initial findings in our preliminary results. And this shows some interesting genes here in this top 10. Particular number 2 is a nitric oxide synthase, and at the bottom, number 10 is HCRA1, which is validated as a stroke, coronary artery disease, and microvascular disease gene in some large, elegant studies recently. Very interesting genes at the top 10 here, and I'll talk a little bit more about those. The top one is HHIPL1, and rare predicted loss of function variations in Pakistani genomic resource that we identified in the study. All of these predicted loss of function mutations had higher odds ratios for myocardial infarction or coronary artery disease, suggesting loss of function was associated with increased risk of disease. We validated this in 2 separate studies, genes and heart, that genes and health, that is also enriched in South Asian population and gives a very similar signal. And then in a unique population in Finland, an isolate population, we also found in a very large case control validation similar positive findings for many of the top genes that we found in our discovery studies, and HIPL1 remained very positive in this resort. And again, in Finland, there was a very common 1% rare variance not present anywhere else in the world, enriched in Finland. And this predicted loss of function variant was also associated with increased risk of coronary artery disease, as we found in our initial analysis. Now, what is this protein? It's hedgehog interacting protein like 1, which means it looks like hedgehog interacting protein, which I'll talk about in a second. And these have a beta propeller, that's a unit in the protein that binds to sonic hedgehog. One previous study suggested that it activated sonic hedgehog and that its knockout in mice models decreased atherosclerosis, which was kind of surprising to us. But the model was that HIPL1 activated sonic hedgehog, and when you lost HIPL1 knockout, you reduced atherosclerosis and you reduced sonic hedgehog signaling. However, this is in contradiction to our human findings, where we have predicted loss of functions with truncation mutations suggesting increased risk for myocardial infarction and coronary artery disease in humans. We are not sure from our data that HIPL1 either activates or inhibits sonic hedgehog yet, so work in progress. So we're very focused on sonic hedgehog signaling and how HIPL1 interacts with the components of that which are found in the cilia of smooth muscle cells and endothelial cell and fibroblasts in the blood vessel wall. This is a major focus of the basic work in the laboratory right now. We've picked on the top mutations identified in Finland and Pakistan to do structure-function relationships and to look at their binding of sonic hedgehog and the beta propeller interacting domain. So this is leading us to really study sonic hedgehog. And I'll come back to that in 1 second, but the main questions we are in the middle of addressing are whether the mutants in HIPL1 reduce secretion of the protein, do they bind to sonic hedgehog, do they have mutants of reduced binding, do they have activate sonic hedgehog or not, or is there sonic hedgehog independent going work? So what have we found? HIPL1 predicted loss of function is associated with increased coronary artery disease and MI risk. We have implicated sonic hedgehog signaling in atherosclerotic cardiovascular disease, but we have to prove that's the mechanism of HIPL1 action. So we're continuing to work on the mechanisms. We're expanding our recall studies in Pakistan for this gene and others that we've discovered in this discovery study. We continue to work on functional studies in vitro, human translational, particularly where is this expressed in the plaques, and we're making mouse models of the human mutations to better understand them in vivo. So finally, to add to the perspectives I presented in the beginning, we have identified that smooth muscle-derived cells have tumor-like behavior, and this might be a therapeutic opportunity in atherosclerosis, particularly in residual risk on low LDL therapies. In addition, not shown here today, we've also been looking at fibroblasts as a stromal cell that's evolved in atherosclerosis, and I've given you a teaser on HIPL1 as a smooth muscle cell and a stromal expressed gene that may be causal in human atherosclerosis. I want to thank my group shown on the left here, particularly Enrique Garcia, Chenyi Zhu, Thomas Mawson, Hui Japan, as I mentioned, and then collaborators at Columbia, Dinesh Salahin, Alan Toll and Ira Tabus, Henry Zhang and Rob Bauer, and then collaborators across the world, Mingyao Lia Penn, Lars Magdafessel, Munich Vascular Biobank, Dinesh Salahin, as I mentioned, in Pakistan, and Minika Koinen Mata in the University of East Finland in Finland. Thank you so much for your time.